So thank you all for coming to the, sorry, I don't have the laser, but I'll be standing here for my image. So I think the science today I'm going to talk about all centers around the question of how did we develop from a fertilized egg to a complex multicellular organism, a human. Well, the genetics can hardly explain this problem because most of us have two copies of genomes, one inherited from mom, the other one inherited from dad, and they're all identical across all the cell types. So the answer actually lies in the different gene activities that leads to different cell types, which ultimately build up to a multicellular organism like us. And the central mechanism underlying the specification of various cell types is epigenetics. And epigenetics refers to chemical modifications happening on the genome, changing gene activities without changing the DNA sequence. And it is essential to regulate gene activity and also build up a whole profile of gene expression to specify a certain cell type. And this process is not only different in different cell types, but also dynamic during aging and development. When the specific process goes wrong, disease can occur, such as a lot of developmental diseases, cancer, and neurodegenerative diseases. So to be able to study this particular process in detail, in high resolution, in real time, we developed a mouse model using a reporter to track a specific epigenetic mark, which is DNA methylation, in single cells in a live mouse embryo. And here we are looking at is a 2.5-day-old, a dish of 2.5-day-old mouse embryos that are developing. And the color of red, green, and yellow represents the level of DNA methylation in the cell. So as you can see, at this early stage of development, there is already a huge degree of heterogeneity among different embryos, as well as among cells within one embryo. And this process is also very dynamic. As the embryos mature into 4.5 days, you see more heterogeneity in the entire embryo, and cells start to cluster, and cell types that have unique signatures of DNA methylation start to form different cell fates. So we are very excited to apply this tool not only to study normal development, but also in complex biological processes such as disease progression or aging, and the interaction between environmental factors and how does that affect the epigenome. With that, I'd like to thank my lab and my mentor especially, the collaborator, Jonathan, who originally had this idea of this reporter, and special thanks to the genetic engineer mouse model team at the Whitehead, who helped me tremendously to generate these mice. And also thanks to Wendy at the tech facility at Whitehead, who taught me how to use confocal microscopes. So thank you.